Yeah, good morning, everyone. So, in our well as in the female reproductive organs, the next step in the reproductive human reproduction of that is the reproductive cycle is the insemination. Okay, the next step is the insemination. Now, what is insemination? It is just the release of male gametes into the female genital tract called as insemination. Okay, it is just the release of male gametes. Okay, so see further. Okay, so when the sperms will be there in the male genital tract, the sperms will be active for around many weeks. Okay, for 3 to 4 weeks they will be active, but once they are ejaculated out of their genital duct, and once they are in the semen, and once they are out of the body of the males, the sperms will be active only for around 48 to 72 hours. Okay, so only for these many hours they will be active once they are out of the body. Okay, that is what. So where they will get released out, they'll get released out into the female genital tract, and that process is called as insemination. How they are getting released out? By means of copulatory organ, that is the penis. Penis is the copulatory organ which helps in the release of these male gametes into the copulatory canal, that is the vagina, and the process is called as insemination. So once the sperms enters into the female genital tract, then the capacitation of the sperms will occur. Okay, capacitation. Now, what is this capacitation? Capacitation is the activation of the sperms. Okay, it is the activation of the sperms, and they'll get activated once they are inside the female genital tract. The first step in the capacitation is the dissolving of the membrane cholesterol. Okay, dissolving of membrane cholesterol. See, membrane cholesterol is a layer made up of cholesterol present above the acrosome. Acrosome is the part of the sperm which is present above the head. Okay, so if this is the head, this is the acrosome. Okay, above the acrosome, the membrane cholesterol will be there. So this layer will get dissolved. Okay, incapacitation, which in turn will weakens the acrosome. The acrosome will get weakened off because of the dissolving of this membrane, and this is the first step in the capacitation. The next step in the capacitation is the dilution of decapacitation factors. Okay, so decapacitation factors, the dissolving of decapacitation factors. Decapacitation factors are present in the seminal vesicles to prevent the sperms from getting activated in the male genital tract itself. Okay, and they'll be there in the seminal vesicles. Seminal fluid, they'll be there in the seminal fluid, and they will not let the sperms to get activated inside the genital tract. See, if they are getting activated in the male genital tract, means then it will be a waste because there won't be any ova. And in our previous classes, we read that ova can't move. Okay, because one thing it will be heavy, other thing there are no locomotory organs for the ova. That is why if they are getting activated in the male genital tract, means they will get, it will get wasted. Okay, that is the reason decapacitation factors will be there in the seminal fluid. So once they are once the semen with the sperms will enter into the vagina, the dilution of these factors will be there, because again say these factors will not allow the activation of the sperms. So that is why dilution is important once they are inside the female genital tract. The next step in the capacitation of the sperm is the entry of calcium ions into the sperms. Okay, so calcium ions will cause us the rapid movements in the tail of the sperm, and tail is the locomotory organ of the sperm. With the help of tail only it will move further. That is what the entry of calcium ions will be there. The sperms will get activated, then the movement of sperms will be there. So how the movement will be there? From where the movement will be there? The movement of the sperm will be there from vagina to cervix to cervix, and then it will move from the cervix to uterus, and from the uterus it will move to the fallopian tube. Fallopian tube. That is the oviduct, what we read in our previous classes. So this is the movement of the sperm once they enter into the female genital tract, and this you should remember. So after capacitation, the movement will be there. The sperm is moving via vagina, and then it will crosses the cervix, then uterus, and finally it will reach to the fallopian tube. In the fallopian tube, the sperm will go up to the ampulla and isthmic junction. Okay. So up to the ampulla and isthmic junction. Okay. So that junction is called as ampullary isthmic junction. What it is called as? Ampullary isthmic junction. Okay. So they'll go to the fallopian tube. In the fallopian tube, it will crosses the isthmus. Okay, and then at the junction of the ampulla in the isthmus, fertilization takes place, which is the third step in the reproductive cycle. Okay, so see, after formation of the gametes, the next step, that is after gametogenesis, the next step is the insemination. It is the release of sperms into the female genital tract. Once the sperms are released, the capacitation of the sperm will be there. So this is all will be happening in the capacitation. Okay, this all will be happening in the capacitation. So after capacitation, the movement of the sperm will be there. It crosses vagina, it crosses cervix, then it will go to the uterus. After crossing the uterus, it will go to the fallopian tube, and in the fallopian tube there will be a junction, that is the part where the ampulla will get continued with the isthmus, that is the ampullary isthmic junction. Here in this junction, the fertilization takes place. And when the fertilization takes place, when the ova will be there. Got it? So the sperm has to move up to here. Simultaneously, at the same time, the ova should be moved. And from where the ova will get released? Ova will get released from the ovum. Okay, so ova will get released from the ovary, and it will be taken up by the infundibulum. So on the infundibulum, fimbriae will be there, helps in the collection of ova from the ovary. And then the movement of ova will be there because of the ciliated columnar cells present in the oviduct. So these ciliated cells, they'll push the ova towards the ampulla. From the infundibulum, movement of the ova will be there towards the ampulla. And after reaching into the ampulla, ova also will reach at this ampullary isthmic junction. At that time only, if sperm is there, means then the fertilization will take place. If there is no sperm, means there won't be any fertilization. The ova will be released out after a period of time, what we read in our previous class, that is in the menstrual cycle. That is why, see, both the gametes should be there simultaneously, should move simultaneously. Both of the gametes should be there at that particular time. And again, see, that is why it is said that not all the copulation or the coitus leads to pregnancy. Now, what is this copulation or coitus? It is nothing but the release of sperms into the vagina. That is called as copulation or the coitus. Another name is coitus. Okay, that is what all the copulation and coitus will not lead to pregnancy, because at the same time the movement should be there. And again, as you people know that the ova will get released on the 14th day of menstrual cycle. If before that 14th day, the sperm is arriving at the ampullary junction, means that no one will be there to get fertilized. That is why there won't be any fertilization. And again, ova will be active for 24 hours. So after 14th day, 15th to 16th, if we are taking also, means after the 17th day, there won't be anyone for the fertilization. If copulation is there also means it will not lead to any pregnancy. That is why it has been said that not all the copulation or the coitus will lead to pregnancy, because the movement of the gametes should be there simultaneously. One, okay, together they should move, both the ova and the sperms. And the next step, after insemination is the fertilization. Okay, the next step is the fertilization. Okay, so fertilization is the union of male and female gametes, and where the fertilization will be there, it is there in the ampullary isthmic junction. Okay, ampullary isthmic junction, the fertilization will be there. Okay, so the next step, that is the third step, is the fertilization, which is followed by the implantation. Let us understand with the fertilization. Okay, so after the movement of the sperm, so the first thing happening while fertilization is the acrosomal reaction. Acrosome reaction or the acrosomal reaction. Okay. So see, basically the definition of fertilization is the union of male and female gametes. That is called as fertilization, which leads to the formation of zygote. Okay, leads to the formation of zygote, which is diploid. We have read, we have discussed previously also, gametes will be haploid, union will lead to the formation of zygote, which is diploid, and that is what we are calling it as fertilization. Okay, so see, once the ova reaches the ovum in the ampullary junction, then the acrosomal reactions will start. Okay, the acrosome will get activated and release the enzymes, the hydrolytic enzymes, or also called as sperm lysins. Okay. Okay, so see, so once the ova and the sperm will meet in the fallopian tube, then the acrosome will get activated. Okay, now what is acrosome? Acrosome is present above the head. Okay, so this is our acrosome. This is our acrosome. So this acrosome will get activated once the sperm reaches to the ova. It has got produced from the Golgi bodies. We have read in spermiogenesis, the Golgi bodies will get modified to form the acrosome and will get filled up with the hydrolytic enzymes. Okay, so these enzymes are also called as sperm lysins, and there will be three types of this enzyme. The first is the hyaluronidase. The first enzyme is the hyaluronidase. Hyaluronidase. Okay, so this enzyme, so dase, okay, is an enzyme. So hyaluronic. Okay, so this enzyme will dissolve the hyaluronic acid. Okay, so what this enzyme is doing, it is dissolving the hyaluronic acid. So where the hyaluronic acid is there, it is there in the ovum. It is there in the ovum. Now where in the ovum? It is there in between the follicular cells. Where it is there? It is there in between the follicular cells. What it will do? It will work as a cementing factor. It will join up the follicular cells together to provide the protection to the ova. But the sperm has to reach the ova. The sperm, it has to reach the ova. Above the ova, follicular cells are there. They're compactly joined together by means of hyaluronic acid. That is why the hydrolytic enzyme, one type of hydrolytic enzyme is the hyaluronidase. This enzyme will dissolve the hyaluronic acid, which acts as a cementing factor between the follicular cells. So dissolving of the hyaluronic acid will be there, which loses the follicular cells. The next enzyme is the corona penetrating enzyme. What is the next enzyme? It is the corona penetrating enzyme. So corona penetrating enzyme will dissolve the corona radiata. Okay, corona radiata of the ova. The another enzyme or the third enzyme is the zona lysine, also called as acrosome. This enzyme will help in the dissolving of zona pellucida. Okay, zona pellucida. Okay. Okay. Zona pellucida. Okay. So as soon the sperm will come in contact with the ovum in the ampulla, acrosomic reactions will get started. The acrosome will releases the hydrolytic enzymes, also called as sperm lysins. There are three types of hydrolytic enzymes produced by the acrosome, which you have to remember. They are very important. The first one is the hyaluronidase. It will dissolve the hyaluronic acid present between the follicular cells. Okay, it helps in cementing the follicular cells together. Compaction of the follicular cells will be there because of hyaluronic acid and will get dissolved by means of hyaluronidase. The next enzyme is the corona penetrating enzyme. Corona penetrating enzyme dissolves the corona radiata surrounded the ovum. Okay, the next enzyme is the zona lysine, also called as acrosine. Okay, zona lysine is also called as acrosine, helps in dissolving the zona pellucida. Okay, further see, as soon the acrosome will come in contact with the ovum, as soon this acrosome will come in contact with the ovum, there will be a formation of a fertilization cone by the ovum. When all this will be happening, no, meanwhile the ova will be producing a fertilization cone. So like this. Okay, this is the fertilization cone. Okay, fertilization cone, also called as cone of reception. Fertilization cone, or also called as cone of reception. Okay, so when the acrosome comes in contact with the ova, this cone formation will be there. When all these things will be happening, no, meanwhile the ova will produce the fertilization cone. Okay, which leads to the other cycles in the ova. Okay, so next let's go to the other changes, that is the corticular reaction and the zona reaction. So let's go to that. Okay. Okay, next go to that. So the next step after all this is the corticular reactions and the zona reactions. Okay, the corticular reactions and the zona reactions. Okay. The next is the corticular reactions, cortical and the zona reactions. Okay, after the formation of fertilization cone. So once the formation of fertilization cone will be there, then the corticular cells, okay, and the zona cells, okay, will get activated. So so once the fertilization cone will get formed, okay, fertilization cone will get formed, then the influx of sodium ions will be there. Okay, sodium ions will be there in the ovum, membrane. Okay, So what is happening after the fertilization cone? Influx of sodium ion will be there. Influx of sodium ions. Okay, influx of sodium ions will be there. See, influx of sodium ions causes the depolarization of the ovum membrane. Ovum membrane will get depolarized because of the sodium ions entering. This depolarization further causes the fast block to check polyspermy. Okay, the fast block to check polyspermy. As soon the fertilization cone will get formed, the sodium ions will enter into the ova membrane, which in turn causes depolarization. In the sense, the ova membrane will get activated. As soon it will get activated, first block will be there to check the polyspermy. So what the ova membrane will do? It will block it so that the other sperms will not enter. Now, what is this polyspermy? Entry of more than one sperm will be blocked by this process. So this is the first block. Okay, so when the fertilization cone formed, when one sperm's acrosome got attached with the ova, because of that the fertilization cone has got formed. So once the fertilization cone will get formed, the influx of sodium ions will be there in the oval membrane, which in turn causes the depolarization of the membrane. The ova membrane will get activated and it will stop any other sperm to take the entry because already one sperm has got entered. Okay, already one sperm has got entered. That is why any other sperm will not enter. That is what called as polyspermy. It will check, it will stop the polyspermy. Meanwhile, the calcium ions will enter into the ova membrane. Okay, into the ovum, the calcium ions will get entered. These calcium ions will cause us the extrusion of the cortical granules. It causes the extrusion of the cortical granules. And further, they also causes the thickening. They also causes the thickening of the zona pellucida. Okay, zona pellucida. So the extrusion of the cortical granules and the zona pellucida, thickening of the zona pellucida will further protect the polyspermy, and that is what we are calling it as slow block to check polyspermy. So block to check polyspermy. So see, the entry of more than one sperm will get protected by means of these two processes. That is, the first block to check polyspermy will be there by means of sodium ions, and then the slow block to check the polyspermy will be there by means of the cortical granules. Okay, that is the cortical reactions, and then the thickening of the zona pellucida. The zona pellucida will become impervious to the further coming sperm. That is why the ova will not get fertilized with more than one sperm. That is what the competition, what we are calling it as. As soon one sperm will reach us, it's over. It's over. No other sperm can enter. That is why the sperm has to be very active, because see, millions of sperms are getting released in one ejaculation, and that one sperm only can enter the ova. As soon the sperm is entering into the ova, all these things will be happening. Finish. No one can enter. Okay, so blocking of the polyspermy will be there. One by means of sodium ions, that is the first block. Another by means of calcium ions, that is by means of extrusion of the cortical granules and the thickening of the zona pellucida, that is the slow block to check the polyspermy. Okay, so this will block. Now only one sperm can enter. Okay, so that one sperm then leads to the formation, that one sperm will get meet up with the ova, okay, nucleus, the ova, and that results in the formation of zygote. Okay, so other sperms can't enter. One ova has already entered, which will get mixed up with the ovum. It will meet the ovum and leads to the formation of zygote. And the formation of zygote is called as fertilization. This is what actually called as fertilization. Okay, and further see, the gametes will be haploid. The union of male and female gamete. The union of male gamete, okay, that is the sperm, and the female gamete, okay, that is the ova, is called as fertilization, which leads to the formation of zygote. Leads to the formation of zygote. Okay, so here the sperm will be haploid as it is a gamete, then ova will be haploid. The resultant will be a zygote. Zygote is also called as synkaryon. Remember, commonly we'll call it as zygote. Another name is synkaryon. Okay. So zygote or synkaryon will be diploid. Because see here, the gametes will be haploid, we have read already. And this union of these gametes to form zygote is called as fertilization. Okay, so see, after the formation of the zygote, so the male and the female pronuclei will start fusing. The male and the female pronuclei will start fusing. So when the nucleus will be fusing, the nuclear membrane will get disappear, and the chromosome will become free. They'll become out. They'll get free into the cytoplasm of the ova. See, the sperm has entered into the ova. We read just now, the sperm enters into the ova. Okay, so as soon it will enter, the pronuclei will start fusing. How they'll fuse? By the dissolution of nuclear membrane. The chromosomes will let free into the cytoplasm. So what happens during that time, during the fusing of this pronuclei, the spindle fiber formation will be there. Okay, spindle fiber formation will be there between the two chromosomes. So one chromosome of father, another chromosome of mother. In between these two, the spindle fiber formation will be there. The spindle fiber will get formed to hold the chromosomes together so that these chromosomes will not get dispersed into the cytoplasm. Okay, so after when the sperm enters into the ova, the fertilization occurs. So while fertilization, the pronuclei fusion will be there. So during this fusion, the nuclear membrane will get stopped, where the chromosomes will be there. So we are talking about the chromosomes here. So that is the sets, the haploid set, the diploid, the haploid sets will get fused to form a diploid, right? So how that will happen means the nuclear, so these chromosomes will be there inside the nucleus. Nucleus will have a nuclear membrane. So that nuclear membrane has to get dissolved. So once the nuclear membrane will get dissolved, the chromosomes will become free into the cytoplasm. They may get dispersed. That is the reason the spindle fibers will get formed. These spindle fibers will hold the chromosomes together. Okay, at a place the chromosomes will get holded up. And then after this, the mitotic division will be there. Because see, zygote is a single cell. So mitosis will be the next step, because from zygote to millions of cells has to get formed, you know, like the complex organisms. We have come out from this zygote only. How means zygote has underwent the mitotic division. So that is what here the spindle fibers will hold them together so that the chromosomes will not get disturbed, and then the mitotic division will start. So during the mitotic division in the anaphase stage, the separation of chromosomes will be there towards the centrioles. We have read this. Okay, so during the separation of the chromosomes at the centrioles, both the male chromosome and the female chromosomes together will segregated at the centrioles together, both. Okay, little of this, little of that will get segregated at the centrioles, which in turn leads to the formation of the the daughter cells. Okay, that is why it is not wrong to say that it is the mitosis after the zygote. It is the first mitosis after the formation of the zygote which actually leads into the fusion of the parent chromosomes together. Got it? So it is the mitosis after the zygote. It is the first mitosis after the zygote which actually leads to the fusion of the male and female gametes. This we have to remember. It is very, very important. Okay. So after the formation, after the sperm enters into the ova, the fusion will get started. Okay. So when the sperm enters into the ova, it leads to the formation of zygote, that is nothing but fertilization. Then the fusion will get start. Nuclear membrane will get dissolved. The chromosomes are now free into the cytoplasm. If they are free, if they are let like that only, means they'll get disturbed into the cytoplasm because they'll be very, very minute. They'll be very small. They may get dispersed into the cytoplasm. That is the reason spindle formation will be there, which will hold the chromosomes together in one place. After the formation of the spindle fiber, that zygote undergoes the mitotic division. So during mitotic division, in the anaphase stage, the chromosomes will get segregated equally at the opposite poles, which leads to the formation of the daughter cell. That is why it is the first mitotic division after the zygote formation which has actually lead to the fusion of male and female chromosomes. This is important for us. And this completes our next cycle, that is the fertilization. Okay, the next step is the sex determination. Okay, sex determination, actually it is illegal. Generally in India it is illegal because of many reasons. It is illegal because most of the people in our country prefers the male child. That is why it is illegal. But see, the sex is actually defined by the male. Okay, the formation of a male or a female child is mainly depends on the male. It is not on the female, but that too the female will be blamed in our country, and that is the reason sex determination is illegal in our country. And generally because see, many people will go with the abortion if the female child is there. Now we'll understand how exactly the sex determination will be there. Okay, see, the gametes produced by the female will be having only X chromosomes, whereas the gametes produced by the male will be having X and Y chromosomes. Means the gametes of the female will be having only X chromosomes, whereas the sperms will be either carrying X chromosome or it may be carrying Y chromosome. So 50% of the sperms will be carrying the X chromosome and another 50% will be carrying the Y chromosome, whereas ova will be having only and only the X chromosome. If the X chromosome, if the sperm with the X chromosome unites with the ova, means it leads to the formation of female child. It leads to the formation of female child. If the Y chromosome of the sperm unites with the X chromosome of the female, means it leads to male child. That is why, see, sex of the child is mainly depends on the father rather than the mother. Mother is not at all involved in the formation of the sex of the child. It is only and only the father who is mainly responsible for the formation of either a male child or a female child, because it is the father who will carrying the two types of sperms. One sperm will be carrying the X chromosome, another sperm will be carrying the Y chromosome. And again, remember, not both the chromosomes. No, not both the chromosomes. either X chromosome or Y chromosome. 50% will be carrying X chromosome, another 50% will be carrying the Y chromosome, sperms. Whereas ova will be having only one X chromosome, only one. Okay, that is what the haploid set, what we'll call it as haploid. Okay, haploid. Single set of chromosome, haploid. Okay, that is what, if the sperm with the X chromosome unites with the ova, means then it will be a female child. Sperm with the Y chromosome unites with the ova, means it will be a male child. Okay, further see mixing of these chromosomes. Okay, so mixing of these chromosomes, that is the mixing of the male and the female chromosome, that is the father and the mother chromosome, is called as amphimixis. What it is called as? Amphimixis. That is the mixing of the father and the mother chromosome is called as amphimixis. Got it? So this is the that is the third step in our reproductive cycle, that is the fertilization. Okay, so fertilization is the formation of zygote because of the union of the male and female gamete. Okay, and before fertilization, many events will be there. The one event is the acrosomal reaction, during which that is the release of 3 types of enzymes will be there. Okay, and then the fast block of polyspermy will be there, followed by the slow block of polyspermy. Ultimately, one sperm will enter, and that one sperm will get fused up with the ova. Okay, pronuclei of the ova will be there, which leads to the formation of zygote, and then the zygote will undergo mitotic division to form the embryo. So the embryo development will be reading in our next class, and this is about the fertilization and the sex determination. See, sex determination is mainly dependent on the male, it is not on the female. Further, sperms will be either X or either Y, not XY. Got it? And the formation, the combination of the chromosomes from the mother and the father is called as amphimixis. Okay, the mixing of these chromosomes is called as amphimixis. So we'll end up our class today, and tomorrow we'll be going with the embryonic development. Okay.